Guys, just a minute. Just hang on. Hey guys, welcome to the class. Good evening. How are all of you? Yeah. Okay. So, before we start, let me just bring you up to the speed. So, in the last class, we started with the very first chapter of biology. That was cell, right? The fundamental unit of life, cell. And I hope you people enjoyed it and you people had certain takeaways. Okay. So, good evening and welcome to the class again. So in this particular lecture, we are going to start with the next chapter, that is tissues, right? Sorry. So I have many students of you who are in our regular batches, and they ask for a chapter overview. So I think this is a great place where you can get all of that and a bit insight into what you can expect in the paper. Akshita, don't worry. Try to log in into the Merit Nation website. You'll find it there. We have the database there itself, so you'll find it. Okay. Okay. So cool. Let's get into the chapter already, right? Okay, so chapters tissues, right? And we have already discussed weightage in the last class, right? So we know that for the biology, you have organization in the living world, which comes of 20 marks. I'm just repeating this so that if someone of you have missed it in the last session, they know this. Okay. Then we have our environment, and then we have food and food production. So as you can see, altogether, 30 marks of syllabus comes from biology portion, right? So do well to remember that biology caters a great part of your exam paper out of 80. So make sure you dedicate ample time for that preparation. Okay, so let's go deep into the chapter. As you can see, it's a huge network right in front of you, right? Raki, you have that on the Merit Nations website. Okay. Oh, no worries, fun time. Okay, guys. So let's try to understand tissues. So here we'll not go into the detail. Rather than that, we'll try to understand the overview. Okay. So tissues. Generally, first we divide the tissues into plant and animal tissues, right? And we need to understand the differences, the core differences between plant and animal tissues. We need to understand that in the plants, the growth is indeterminate; it happens throughout the life cycle, whereas in animals, it's limited to a certain stage, right? Plant tissues are generally made up of dead cells or largely made up of dead cells because they largely need structure. Plants don't move around, they don't locomote, so they don't need much of movement when it comes to locomotion, right? So they are largely made up of dead cells, not all, but very large chunk is dead. Whereas animals, we are largely made up of live cells, right? So who will quickly tell me what's the part or the organ where we have dead cells in animals? In animals, what's the part or the organ where we have dead cells? And I want at least one of you to give me the correct answer for this. No, in animals, Akshita, not sclerenchyma. Hair, right? Hair and very importantly, skin, right? So the top layer of the skin is dead. Nails, as nails too, right? Fur counts as hair, Shubham, so same thing, right? So then let's divide the plant tissues into two types based on the growth. So meristematic tissues are the ones which have the potency to grow, to divide, right? And we divide them on the basis of the position: apical, that means on the apex; intercalary, that means in the mid; and lateral, that means on the sides. Srivastav, you were there in the 10th session as well, I remember, I think. Yeah, so cuticle has a substance called cutin that provides a waxy coating and prevents the loss via transpiration. Okay. Vardhan, muscle fiber, that's animal cells. We'll discuss that in brief when we come to that. Okay, so come on, guys. Permanent tissues, that means which lose the capacity to divide. We have simple and complex. Simple has only one type of cells, and complex has more than one type of cells. That is one thing that people get confused about and mess this up in the exams. Okay, so simple can be again divided into parenchyma, collenchyma, sclerenchyma on the basis of their function, their structure, their cell wall. You must be very well aware about how they look in the under the microscope. Okay, so you might directly get questions. So the key feature is that parenchyma has thin walls, Collenchyma has uneven walls. Okay, and then sclerenchyma is mostly walls. Okay, so the cells are of sclerenchyma are dead because they are generally there for support. Deepak, a very good question. They are growing because the base has the cells that divide. Okay, the base is growing, not the top part. That is why the length increases because the base at base they are growing. Okay, yes, Akshita, that's correct. So then we come to the complex permanent tissues, which are made of more than one type of, cells. So we have xylem and phloem, right? Xylem has the job of conducting water and minerals, whereas phloem has the job of conducting food, right? Uh, then xylem, we have tracheids, xylem vessels, parenchyma, and fibers. Very important. Tracheids and vessels. The differences between tracheids and vessels are important. Tracheids are pointed, long, whereas vessels are wider, but they are a bit shorter when compared to tracheids. Right? They are not, Deepak. How come it's they are not? Otherwise, there won't be any growth. See, at the base only they have some extent of vascularization, Deepak, right? Isn't it? So at least some blood vessels will come there and give them the nutrients at the base, the very top layer, the very top part, not exactly the top layer of skin, but a bit deeper, right? So that is where they get the nutrition, and hence they are not dead. Okay, sclerenchyma are dead, first of all. Okay, now xylem is mostly when you compare xylem, we have mostly dead cells, whereas the fibers are living. Okay, and the vice versa is true for phloem. So you need to know the differences like that. Now, phloem has sieve tubes, which will do the work of transport. Important thing here is the sieve tubes, they don't have a nucleus. It's very frequently asked in exams as a one marker sometimes. And companion cells have the nucleus. So all the needs that the sieve tubes have is fulfilled by the companion cells. Okay. Then we have the phloem parenchyma and fibers. Kesha will be doing a question round. Don't worry about it. Okay, we have plenty of time according to our plan, so we'll do that. Minerals, see, generally, Akshita, minerals are dissolved in water. That is why they are transported along with water. When we talk about food, we generally talk about sap. Okay, so food, see, minerals cannot be synthesized, right? As in, like they are present in the soil, they are absorbed along with water. They are soluble mostly, and they're transported via xylem. When we are talking about food, we're talking about carbohydrates, proteins that are made in the plants itself. So the leaves, they make the glucose, right? They capture carbon, and via several metabolic processes, they are converted into everything that the plant needs, right? And that is how it's transported. Uh, Archana, see, the best thing is remember the flow. And that is why we have provided with you with a mind map. So you can take a screenshot of it later, and this will help you to recapitulate the whole chapter. Okay, God of plants can't move with because they could not evolve like that. Companion cells, Chavi, are cells that have nucleus. They are beside the sieve tubes, and they have the nucleus, right? So they will provide all the necessary nutrition that is needed by the sieve tubes. Function of lymph. Lymph acts as a medium of exchange, and more importantly, it transports fat. It also transports several immune cells of the body. Okay, now let's try to understand the animal tissues. We have divided the animal tissues on the basis of the functions, right? We are stopping here to discuss Raki's doubt, how the dead cells divide themselves. No, no, no. Raki, dead cells cannot divide. Only the living ones divide. Only the some cells at the base of it, they are living, and that's why they are dividing. Okay, sap is the fluid part that contains the food materials of the plants or middle. Okay, now epithelial. Epi means on the top. So epithelial generally consists of the tissue that are the coverings, external and internal coverings of the body. We have different types based on the shape. We have squamous, we have columnar, we have cuboidal. Squamous can be simple or stratified. Very good question for many exams, right? So simple has just one layer, stratified has many layers, right? And the placement depends on where is what is needed where. For example, if you imagine your stomach, right? Or let's not imagine stomach, let's imagine esophagus. So esophagus has a lot of slugging to do, right? The food passes it, it will erode certain layers. Or if you imagine the anus, so a lot of slugging happens there. So stratified there makes sure that the upper that even after the erosion, the cell is, the layer is intact, the covering is intact. Whereas simple squamous is in the region where we have absorption needed, for example alveoli. Okay, cool. uh, so that is where epithelium is found, Jeanette. So all the coverings of your body, the top, the skin, it's found in the internal coverings of the gut, like intestine, it's found in stomach, it's found in esophagus, everywhere, all the coverings. Next, we come to the connective tissue. So connective tissue basically is a tissue that connects. Okay, translocation happens amridal via the sieve tubes. Okay, now we have connective tissue proper, that is loose and dense. We have areolar, adipose. Areolar has air spaces, adipose stores fat. Tendon and ligament connects the bones and muscles. How? So this is how I remember, because people generally forget. So BTM, bone, tendon, muscle, and BLB, bone, ligament, bone. So a ligament joins two bones, and a tendon joins the bone and the muscle. Ciliated. Imagine, Uday, where will be the ciliated epithelium be required? Where cilia lashes, right? So it moves. Where will the movement be required? It will be required in the esophagus to some extent, where they have to move the mucus, right? To some extent, it will also be present in the trachea, right? There they have to move the substances, right? So that is there where they are. Is bone a connective tissue? Yes, Gaurav, it is in fact a connective tissue. We'll come to that, bone and cartilage, don't worry. So next we come to the fluid connective tissue, which are two, blood and lymph. Now the very important part is blood, right? So blood has cellular part and non-cellular part. The cellular part comprises almost 45% of the blood, which is the red blood cells, the WBCs, and the platelets. Okay, now remember, platelets are not exactly cells, they are cell fragments. Okay, so, and you must remember the function of each. Red blood cells carry oxygen, WBCs are for immunity, and you can also say that platelets are for the clotting of blood. Right. Uh, then you have to come to the skeletal tissue where we discuss bone and cartilage. So the first difference comes in the cells. So the cells of bones are osteocytes. The cells of cartilage are called chondrocytes. Bone is more stiff, more rigid. Cartilage is less stiff, less rigid. Bones have the matrix of calcium phosphate. That is what gives the strength that it has. Cartilage, on the other hand, is largely made of chondroitin sulfate, so it's less rigid. Is platelet also a connective tissue? Platelet is a cell. Shavi, don't get confused. Platelet is a cell that is present in blood. Blood is the tissue. Tissue is an aggregate of cell that has the same function, right, Shavi? So that is what it is. Oh, great, thank you, Midul. Tripti, plasma is a fluid part of the blood, right? The water content, the proteins dissolved in it, the glucose, the waste material, everything is plasma. Except the cell, everything is plasma. What is matrix? Have you eaten raita, Gaurav? Have you ever eaten raita? Yes. So in the raita, there's a fluid part, and there are certain pineapple pieces. So the pineapple pieces are your cells, and the raita is the matrix. So a matrix is nothing but the continuity of the fluid, the gel which surrounds those cells. Okay, and as you in the raita you have the curd generally, right? That is how. So bones and cartilages, they have their own functions. Bones generally are used for the movement of muscles, right? Cartilages have several functions. First of all, they are excellent connectors. So you'll find cartilage attaching or several bones. For example, your ribs and your the breastbone are attached via cartilages. Okay, then cartilages are also present in ears, for example pinna, right? So there also it helps in some sort of temperature regulation as well. Osteocyte and chondrocyte, Akshita. Osteocyte is bone and chondrocyte is um, cartilage. um, Yappan, lymph is connective tissue that is there in the lymph vessels. Like you have blood vessels, you have also lymph vessels. Now there it has certain jobs. Like the lymph vessels, they extract, they take in the fat, they transport it throughout the body. Keshav, plasma is what contains everything, every fluid part. For example, proteins are transported in the plasma, glucose is transported in the plasma, hormones are transported in the plasma. Circulatory system has two circulatory fluids. Blood is one, lymph is another. Blood is circulated by a heart, via heart. Lymph not by heart, rather by other mechanisms. Chavi, you don't need to know the name, but okay, ear cycles, they're collectively called ear cycles. If you want the individual names, it's Malus, Incus, Stapes. Let me write it right here. If you have Malus, Incus, stapes, the three smallest bone of our body. Now let's come to muscular tissue. Parts of the ear, Aryan, this is really not in your scope right now. That is why I'm not going there. Okay. Muscular tissue has three types: striated, smooth, and cardiac. Striated, that means they will have striations, the bands of dark and uh, Ninad, see, because this is here we basically give you the exam strategy. That's why I'm not discussing it in detail here. Okay, so if you're in one of our batches, we'll definitely talk more about it there. So striated generally controls the skeletal muscles. That means the voluntary striated is also known as skeletal muscles. So they are generally used to control the voluntary muscles, where smooth is used to control involuntary muscles, and cardiac is muscle is only found in the heart, right? And there it is used to create the contractions and relaxations of the heart. And the last but not the least, we have the neural tissue. Neural tissue is made up of neurons. Some things that generally people forget is the neuroglia. Okay, so neuroglia is another type of cell that is present in the nervous tissue. Voluntary, which is under your control, Cody. Okay, so neuroglia and neurons, they together make the nervous tissue. So that is the summary of all that you need to know at this level. Yashvardhan, cyton is the upper expanded part of the neuron, which receives the impulse from the dendrites. From there, it transports it to the axon, and axon gives it to the axon terminal. Okay, dendrite web of, just let me draw a neuron. I'll try to draw a neuron here, but I'm not sure how successful I can be in doing that. Okay, so it's not a very fair drawing right now, but see this. Okay, so yeah, let's try to understand. So this part, this upper expanded part is the cyton, and this part is the axon. Further, these endings that you see here, these are called dendrites. They receive the impulse, right? and they transmit it to the cyton. Cyton also has the nucleus. Then the impulse travels all through the length of axon to these places which have the axon terminals. These axon terminals meet either other dendrites, the dendrites of a new neuron. Okay, so these might meet a new neuron, or it might this junction is called synapse. Okay, so it might meet a new neuron or a muscle where the impulse is passed, the information is passed from the neuron to the muscle. Okay, so that is how it operates. Let's go and discuss next. So important topics. What should you be knowing in this chapter? So structure of leaf, very important. You can be anytime asked about the stomata, the epidermis, the parenchyma. Okay, then placement of animal and plant tissues is very important. What is present where? Especially asked is epithelial tissue, collenchyma. Okay, they are very frequently asked questions. Diagram of striated versus non-striated. You can be asked to recognize, you can be asked to draw. Diagram of a neuron, very important. Most frequently asked. Recognize the and label the xylem. So you should be able to label the xylem. Okay. A BTM is that's a way to remember Yapam. BTM, I remember that tendon, they connect bone to the muscle. So B for bone, T2 tendon, M muscle. B2B, that is bone. Actually, BLB is bone, ligament, bone. That is how I remember it. Involuntary muscles are present in your stomach. You can't regulate it, right? Those are involuntary muscles. Is color of nerve cells fixed? No. See, nerve cells, they generally appear white. The axon part appear white. The cyton parts appears a bit grayish. Now you can be practical based question can be related to the plant and tissues, animal tissues under a microscope, right? So you can be asked any question related to that. Now let's practice a few questions. Walls of capillaries are made up of simple squamous epithelium. Why is it the appropriate choice? So who will give me the answer to this question? Come on. So why is it made of squamous epithelium? So it's made up of squamous epithelium because there it needs to there the oxygen, the glucose needs to pass from um, the uh, the capillaries outside so that it can diffuse to the tissue fluid and then to the cells. That is why they are very thin, right? Now, what subtype of permanent tissue manufactures food? Now let's go back and try to understand the permanent tissues in this question, right? So permanent tissues, we have simple permanent tissues and parenchyma. So parenchyma is actually the one that uh, makes food. Actually, there's pellicid parenchyma and spongy parenchyma. You remember studying this when you study the structure of the leaf. Then 4 components of phloem. You should remember that we just discussed it, right? And name the receiving and the transmitting ends of neurons. So receiving end is a dendrite, as you have answered correctly, and the transmitting end is the axon. Chlorenchyma. Okay, see, Vardhan, chlorenchyma is just a special name given to a parenchyma, okay, which performs photosynthesis. So that is correct. Even chlorenchyma is correct. Or you can simply say pellicid parenchyma. Even that is correct. Okay, that is correct. Now differentiate between bone and cartilage. As I just talked a bit in the while discussing, the differences are the name of the cells. That's not a very key difference. The key difference is that bones are generally hard, cartilages are soft. Bones have a matrix of calcium and phosphate, whereas cartilages have chondroitin sulfate. Right? So you should be able to understand the differences between these two. Okay, so are we clear on this? Which parenchyma present? Parenchyma is generally present in the internal layers of the leaf, such as pellicid parenchyma and the spongy parenchyma. I don't have a picture with me here per se, but that is where it's found. Okay, now practical questions. Very important, guys. Where's cartilage? What is cartilage? Cartilage is another tissue. Cartilage is another tissue which is generally present, sometimes joining the bones. Okay, I will try to improve it a bit. Ayapam. See, it's generally present while joining the bones. For example, ribs, attaching ribs to the sternum or the breastbone. Akshita, the entire session is available for on YouTube. You can watch it, you can download it, you can take a screenshot. It's your wish. Okay, now let's quickly discuss these diagrams. Now we have been given the transverse sections of a cell. Now you have to differentiate between collenchyma and sclerenchyma. Okay, so as you can see, the thicknings, the wall thicknings are uneven, right? You can see here the thicknings of the wall are uneven. So this is how a collenchyma looks like. If this had been sclerenchyma, it would be almost filled with cell wall and nothing else. So if it would have been sclerenchyma, it would be almost filled with cell wall and nothing else. Okay, so that is how we differentiate. So that is a key difference between collenchyma and colon and sclerenchyma. They're simply cell wall, Akshita, nothing else. They're simply cell wall. Okay, okay. So guys, that brings us to the end of the discussions. There's practical questions as well. Now see, here we are asked to differentiate between striated, unstriated, and cardiac tissue, right? So striated, as you can see, right? So we have rectangular cylindrical cells, and we have striations. We have dark zones and light zones, right? So these are called striated muscle cells. They generally work for voluntary or muscles that are under your control, right? Whereas the smooth muscle cells are generally spindle shaped. Can you see the cells are spindle shaped? Right? They're spindle shaped. Further, they generally work for involuntary muscles. And then comes the cardiac muscle cells. Now, the key characteristics of cardiac muscle cells are they're branched. They're branched and they have these discs in between the cells, right? They are present where? They are present in the heart, where they do the job of contraction and relaxation in the heart, right? So you should be able to draw these. Very important. You should be able to draw these and lay out the differences. Very important. So if you are able to do that, you can score full marks in these questions. Now, what should we follow while we are answering our questions? See, this particular chapter is filled with several diagrams. So make sure you draw diagrams very neat. Now, while drawing diagrams, make sure that you label on the one side. Always box your diagram. So if this is your diagram, always box it and give a title. Give a title. Do all the namings on only one side of the picture. So don't label everywhere. Only label at on the one side of the picture. Follow this while drawing diagrams. So, and if there are how many types, let's say how many types of permanent tissue or how many types of epithelial tissue, make sure you're using flowcharts. Okay, examples. Whenever you are talking about a tissue, make sure you're telling where they are generally found. Okay, that helps. And you can practice questions. So we are also giving a top 20 questions for each chapter to you from in the links that are provided to you on the chat, and as well practice from several books or materials that are provided. Okay, so with that, I would like to end the class, but last question that I should like to answer is glandular epithelium. Shivam, glandular epithelium is when the epithelium contains a gland and acts as a secretory epithelium. So it starts secreting, right? So that happens in stomachs. They have glandular epithelium because they need to secrete HCl, certain enzymes and stuff like that, right? So there we have the glandular epithelium. Okay, guys. So we're closing in now for this particular discussion. Don't worry if your questions are still unsolved. You can always contact us for our respective batches. We'll be answering that. Our natural resources. Shubham, we are following the NCRT schedule. Sorry, NCRT time content. Okay, so as it comes, we'll answer that. Okay, okay, guys. Well, that's me and signing off with you guys. See you again in the next session. It was lovely. Bye bye and prepare well. All the best.